Pawan.
Good afternoon, everybody. Now I'll be presenting an innovation, a self-brewed side port assembly to convert intravenous cannula into nerve stimulator needle. Introduction. The success of a peripheral nerve block depends upon injection of a local anesthetic agent in the vicinity of the target nerve or the nerve plexus. Several methods are used to locate the nerve. Electrical nerve stimulation technique using insulated needle is widely practiced since current density focuses at the tip. The disposable coated needles, their availability and affordability limits usage but encourage reuse after re-sterilization in economically weaker regions. Coming to materials and methods, we have devised a nerve stimulator needle assembly with readily available intravenous cannula with side port, spinal needle and 10 cm extension tubing. We have used it for supraclavicular, interscalene, infraclavicular and axillary brachial plexus blocks. The procedure is done under strict aseptic precautions. The inner needle of the 20 gauge cannula is removed. Then a 23 gauge spinal needle is pierced through the inner stem of the white stopper, which is removed from the hub of the cannula. The stopper is fixed to the side port. Then the spinal needle is pierced through the side port, enters the lumen of the cannula and touches the opposite side of the lumen. When the inside needle is reintroduced, it rides over this inside part of the spinal needle and will be firmly in contact with it. The stillet of the spinal needle is partially drawn out and the needle is bent sideward. The stillet is used as a contact point for nerve stimulator with the help of a crocodile clip. This ensures good electrical conduction right up to the tip of the inner needle. The port of the inner needle is connected to a pre-filled 10 cm extension line to deliver the local anesthetic. This is a picture showing our final innovative assembly. The brachial plexus block by different approaches was performed using stimuli of 0.5 milliamperes current at 1 Hz. The isolated movement of the fingers or flexion extension at the wrist ensured correct localization. We have conducted supraclavicular, interscalene, infraclavicular and axillary brachial plexus blocks with the sample size required was 50 adults for a success rate of greater than 90% with an error of 10% for 95% confidence interval in indicated cases of orthopedic surgery of the upper limb. However, we have conducted this study in 162 patients. This is a video showing the supraclavicular brachial plexus block being performed by our innovative assembly. Here we can see the isolated movement of the fingers indicating correct localization of the nerve. Now we are injecting the drug at 0.5 milliamperes of current, again indicating the correct localization of the nerve. The results are as follows. Total 162 patients, the males were 106 and females 56 in the age group of range between 18 to 80 years, and this table shows the type of the block and the number being conducted. Total there were 10 failed blocks, out of which there were 3 were due to faulty localization, 2 were due to accidental artery puncture and 5 were due to patchy analgesia, all in supraclavicular brachial plexus block. The success rate with our innovation was 94%. Coming to discussion, a modified 2He needle has been used for continuous plexus block, but its size and difficulty in sterilization are its disadvantages. An indigenous version of the insulated needle has been devised using 2 IV cannulae. However, the Teflon sheath has to be cut in this version and sterility may also be an issue here due to proximity of the crocodile clamp. Bevel configuration has been blamed to influence nerve injury. However, the immediate and long-term effects of short and long bevel needle impalement of the rat sciatic nerve have been studied. The results suggested that should a nerve fascicle become accidentally impaled during regional anesthesia, The lesions induced by short bevel needles are more severe, more frequent, and take longer to repair than those induced by long bevel needles. The success rate of 94% with our innovative low-cost stimulator needle is inconsistent with the published studies. Limitations. For deep situated targets like sciatic nerve, the length of presently available cannula may not be sufficient. Coming to conclusion, this low-cost assembly using readily available intravenous cannula with side port, spinal needle and 10 cm extension tubing may be safely used as an alternative to the commercially available coated nerve stimulator needle in indicated cases of superficial plexus block. Thank you.
An insulated needle as well.
But
the attempt to assemble an indigenous insulated needle was definitely commendable.
Thank you. Thank you, sir.